I wanted to thank particularly Grażyna for putting together the program for next year. It's so wonderful and exciting. So we're tremendously grateful for keeping us with the new schedule for upcoming year. Today is a very special in one way day, but it's also a very sad day. But I will just start with the positive. We have Janet Sparrow, who is one of the top leaders in our field, working on visual processes in RPE and photoreceptors. And so this is really starting the year with the top-notch scientists, and we're very delighted. Janet was actually a reviewer of PNAS paper of our student, Mr. Hong. And so I thought it would be very appropriate for him to give introduction. John?
Hello, my name is John. I'm an MD PhD candidate in the lab of Dr. Scott Palczewski. It is my pleasure to introduce Dr. Janet Sparrow and Anthony Don, Professor of Ophthalmic Science in Ophthalmology and in Pathology and Cell Biology at Columbia University. Dr. Sparrow did her PhD training in neuroscience at the University of Western Ontario, London, Canada. She then did a postdoctoral fellowship at Cornell University Medical College, followed by another fellowship at Rockefeller University. Her research focuses on bisretinoids that constitute the lipofuscin in the retinal pigment epithelium. Her work has since uncovered mechanisms by which these bisretinoids are formed and exert their deleterious effects on photoreceptor and RPE cells, of particular importance in the context of retinal disorders, including retinitis pigmentosa and macular degeneration. She has been exploring small molecule and gene therapy approaches to reducing the accumulation of bisretinoids. Her work has contributed to also clinical interpretations of fundus autofluorescence images. She has received numerous honors, including Arvo Gold Fellow, American Academy of Ophthalmology Achievement Award, the Alcon Research Institute Award, RPB Senior Scientist Award, Research to Prevent Blindness Award, and the Lou R. Wasserman Merit Award. She has well over 200 publications and has been invited to speak at numerous lectures, including at Arvo, Vision for Children World Symposium on Ocular Albinism, UCSF, Yale, Weill Cornell Foundation Fighting Blindness, American Academy of Ophthalmology, and more. Without further ado, it is our pleasure to have Dr. Janet Sparrow present her talk for us today.
Thank you, John, for the very kind introduction. Before I begin my talk today, though, I would like to acknowledge the sad news that Sam Jacobson died yesterday. Many of us learned this through an email from FFB, and I'm sure that Sam was known to all of who are participating today.
So I was just glad to see Arthur with us today. Maybe he wanted to add a few sentences here. Sam was an incredible collaborator and scientist and just a wonderful human being. I remember just a very quick story when we started working on gene augmentation, very, very early on. He invited me to come to Philadelphia to discuss the project, and he insisted that I stay in his house. And what was really more amazing, he emptied his bedroom, his real bedroom, so I could stay in his place comfortably. This was the kind of guy. He came to this meeting late because he was helping somebody who lost conscious in front of the building somewhere in a medical school. So this was a Sam, very intense, wonderful scientist and collaborator. He will be tremendously missed. I'm just processing this information I learned this morning that we lost a real giant in our field that bridges the clinical work and basic science. Arthur, you wanted to share your thoughts for a second?
Yeah, I will take just a second. I don't want to take Janet's time. Yeah, he loved you guys all, that's for sure. And it's been 2 days. It's been quite shocking. It was very quick. And we are all who spent time day in and day out for 35 years with him, trying to deal with this huge loss. Big shoes to fill. He will be definitely missed, that's for sure.
Hey, Janet?
Okay, thank you, Arthur. Whoops, I accidentally advanced. Well, it's okay, I can start.
Dr. Sparrow, can you share your screen again?
Oh. Is it okay now?
No.
Nope.
No.
I can hear you.
I'm not sure why. Gee, it was fine before.
Just redo the sharing. Click the share screen and then.
Whoops. Okay. Should you do the sharing?
No, you have to do the sharing. I can't.
Okay. Okay. Wow. I don't know why.
Yeah.
There you go.
Okay. Now it's okay?
Yep.
Okay. Good. So today is also January 6th, which is a day that's notable in US history. And so I think we should just begin. So all of us, that includes you and I, we all accumulate autofluorescent material in our retinas with age. This autofluorescence constitutes the lipofuscin of retina. And the autofluorescence that you see in the retinal pigment epithelial cell layer here is the autofluorescent material that I'm referring to. The nuclei are stained with DAPI. So these fluorophores form as a byproduct of the visual cycle. And they also serve as the source of short wavelength fundus autofluorescence, also referred to as blue autofluorescence. And so the autofluorescence that you see in the fundus here, and that can be acquired, for instance, with a Heidelberg spectralis, originates from the molecules in the fluorescent molecules in the RPE. So I'm going to talk about some of the work we've been doing over the years to understand the composition. We've isolated and characterized a number of these fluorophores. I'll also talk about mouse models, some mouse models that we've used. We isolate these bisretinoid compounds in human retina, and I'll also talk about the oxidation of these molecules via both light and iron. And then finally, I'm going to talk about some findings with respect to short wavelength fundus autofluorescence. Before I go any further, although I'd like to acknowledge the many collaborators I've had over the year, particularly at Columbia University, Koji Nakanishi and Stephen Sang, at Harvard, Francois Delory, at Penn, Josh Naif. And then there's many students and fellows that I need to acknowledge. For each of the slides showing data, however, I've indicated the people that participated in the publication. So what are bisretinoids? So these are some of the bisretinoid molecules that we have that we have studied. Many of you will be familiar with A2E up in the left-hand corner here, but these other fluorophores are also present. You'll notice that there are similarities in their structure. They each have 2 arms, and each of these arms originates from a molecule of retinaldehyde. But you'll notice that there's differences in the ring structure at the core of the molecules. A couple of them have these pyridinium rings, and others have other molecules have different structures. And then the last thing I should mention is they all have 2 absorbances, one in the UV from the shorter arm, one from the longer arm, which is an absorbance in the visible range. And of course, it's this absorbance is important because these are the wavelengths that are transmitted to retina. So these bisretinoids or vitamin A aldehyde addicts form non-enzymatically. They form randomly. They have no function. They can form both from 11-cis retinaldehyde and all-trans retinal. They initially a single retinaldehyde molecule will react with phosphatidylethanolamine in the outer segment membrane. So you should note that the formation occurs in the outer segment primarily. Whoops. And reaction with a single retinaldehyde molecule generates this shift base, NRPE. And then if and when a second retinaldehyde molecule reacts, you get these bisretinoids that are not reversible. And NRPE, of course, is a shift base that can undergo reverse, can be reversed. And so these molecules form in outer segments and are deposited in RPE cells secondarily with the material that is phagocytosed by RPE. So they form in photoreceptor cells, accumulate in RPE to the processes of outer segment phagocytosis, and they accumulate in RPE lysosomal organelles. So the role of phagocytosis is often misunderstood with respect to bisretinoid lipofuscin. It's not necessary for the formation, phagocytosis is not necessary for the formation of these fluorophores. If you look at the bottom panel here, you can see this orange colored ribbon approximately at the arrow, and it's interrupted by the blue nuclei of the RPE cells. So this ribbon-like monolayer are the RPE cells, and they contain the fluorescent molecules within the RPE. If you look up at the top panel, this is an image from a MERTK knockout mouse. Instead of being within RPE cells, the nuclei of which are along the bottom of this fluorescence, instead of being within RPE cells, the autofluorescence is within the subretinal space. And so these bisretinoids constitute the outer segment debris that is accumulated in the presence of MERTK deficiency. If it has occurred to you that there might be a lot more material in the MERTK situation versus the wild type, you're correct. When we've measured these, the bisretinoids in the MERTK versus the wild type, there's more of this material in the MERTK, and I may come back to that in a few minutes. So why do these bisretinoids accumulate in lysosomes? Well, a number of years ago, it was suggested by Aldred and Lasky that that the major fluorophore, which turned out to be A2E, had this structure, and the structure shown here, and they also suggested that the molecule accumulates or acquires this positive charge within the acidic environment of the lysosome. It was subsequently shown that, in fact, this structure was incorrect. The structure was revised to this pyridinium molecule, and the charge on the nitrogen in this pyridinium molecule is permanent. So it doesn't acquire that positive charge in the lysosome, and it doesn't deprotonate or protonate. And in fact, it's a neutral molecule because of its counterion, the chloride. So A2E does not undergo a change in charge, and yet it still accumulates. These other two bisretinoids also indicate that a change in charge is not necessary for the accumulation. Both of these molecules, all-trans-retinol dimer and A2-DHPPE, are uncharged, and yet they accumulate in RPE lysosomes. The all-trans-retinol dimer has a cyclohexidine ring that is not charged, and A2-DHPPE, a dihydropyridine ring. It's also been suggested that in the presence of bisretinoid, lysosomal enzymes are inactivated. Well, we know of two examples where, in fact, the enzymes are quite active. In the case of, in the case of A2PE, phospholipase D can cleave A2PE to release A2E. It cleaves at this phosphodiester bond, and A2E is released. We don't detect A2PE in the lysosomes of RPE cells, so we're assuming that this cleavage is occurring in RPE cell lysosomes. And then we've also characterized another form of A2PE that is cleaved by phospholipase A2. And the cleavage here is at the ester bond at position 2. And this leaves a molecule which has a single fatty acid chain. And we call this lyso A2PE. And similarly, we don't detect this in lyso A2PE in neural retina or photoreceptor cells. We only detect it in RPE, so we think this is occurring in the lysosomes of RPE. It's been suggested, in addition, that the proteins constitute the lipofuscin of RPE, and that the proteins may accumulate because of inactivity of the enzymes. But several years ago, we did some work with John Crabb and Joe Holyfield and found that only 2% lipofuscin granules, which are organelles, contain no more than 2% amino acid. Yet we knew that the lysosomal fraction must be good because we detected these bisretinoids in the fraction. So we concluded that any content of protein was minor. So bisretinoids don't seem to be acted upon by lysosomal enzymes sufficiently to degrade them, but they are photoreactive and undergo photodegradation. So this slide summarizes work from a number of studies. I've shown here on the left 2 examples of bisretinoids, all-trans-retinol dimer and A2E. And then in the presence of light and oxygen, these bisretinoids can act as photosensitizers, generating singlet oxygen and superoxide anion, which then go on to photooxidize the molecules. And these oxidation sites are unstable. And so they then go on to cleave, they undergo photocleavage, releasing a mixture of reactive aldehydes and these 2 dicarbonyls, glyoxyl and methyl glyoxyl. So we've detected these dicarbonyls, methyl glyoxyl and glyoxyl, in mouse Brooks membrane. And both Jim Handa and John Crabb have reported several years ago that proteins modified by glyoxyl and methylglyoxyl are present in the drusen under RPE cells. So I suggest that the most parsimonious explanation would be that they have been generated within RPE cells and have penetrated to the to incorporate into sub-RPE drusen. So we wanted to also know whether these photooxidation processes were ongoing in vivo. After doing many in vitro studies, we undertook this project in mice, and we studied mice reared in the dark versus cyclic light. And on the left here are, we used albino C57 mice. And we found that in the mice that were raised under cyclic light, levels of A2E were lower than in the mice raised in darkness. In the albino, actually, the eye receives considerably more light than in a pigmented mouse. And so the intraocular light could explain the reduction in A2E relative to the mice housed in darkness. In the second experiment, we housed the mice, all the mice in cyclic light, and then compared black to the albino mice. And again, in mice receiving more intraocular light, i.e. the albino mice, A2E levels were lower. So we have photoreactive molecules, and so it seemed important to test their potential role in light damage. And to do this, we compared mice having different levels of bisretinoid. And so this would be, for instance, wild type on the left here versus ABCA4 knockout on the right. And we compared 2 ages. Since these molecules accumulate with age, we compared mice that were 2 months old versus 5 months old. The light was delivered only to superior hemiretina, and I've written the conditions here. And so under these light conditions, in the ABCA4 wild type mice at 2 months of age, we didn't observe a lesion. At the same age, we observed a lesion in the ABCA4 knockout mice. Then with the comparison between the 2-month-old and the 5-month-old mice, you can see that we obtained a small lesion with the 5-month-old mice, and then in the 5-month-old ABCA4 knockout, a much larger lesion. So our conclusion from this was that bisretinoids that are photoreactive molecules can be responsible for retinal light damage. So it turns out that bisretinoids are not only oxidized by light, they can also be oxidized and degraded by iron-mediated formation of hydroxyl ion, radical, I mean. And this is, of course, the Fenton reaction where iron and hydrogen peroxide generate this highly reactive hydroxyl radical. So we didn't really set out looking for this. We kind of fell into it from other experiments that we were doing. But what we found is that when we treated mice with this iron chelator, this is a clinically approved chelator, the ferroprone, we found that bisretinoid accumulation was increased. And you can see these measurements below in the panel. So there was an increase with treatment of the iron chelator to reduce iron levels in the RPE cells. We also measured by fundus autofluorescence in the mice, and we observed the same. Fundus autofluorescence was increased in the mice treated with the iron chelator. We observe, and I'll go into this a little bit later, we observed that in ABCA4 knockout mice, there's a thinning of the outer nuclear layer that we can detect at 8 months of age. We found that with the DFP treated, in the DFP treated mice, there was an attenuation of this ONL thinning. So the reduction in iron attenuated the ONL thinning in the ABCA4 knockout mouse. So what about the opposite scenario? What if we were to increase intracellular iron? We looked at this using mice deficient in ceruloplasmin and hephaestin. Josh Danaev created these mice, and ceruloplasmin and hephaestin are peroxidases that oxidize ferrous iron to ferric iron so that the iron can be exported and taken up by serum transferrin. And with the deficiency in these peroxidases, intracellular iron increases. And what we found was in the mice that were completely deficient of ceruloplasmin and hephaestin, A2E levels were considerably reduced. So high intracellular iron was associated with a reduction in bisretinoid, suggesting again that iron-related mechanisms were degrading bisretinoid. This is a mechanistic study we did where we use this in vitro assay where we took A2E, exposed it to iron and hydrogen peroxide, and you can see the combination greatly reduced the amount of A2E that we measured by HPLC. So because we had observed this difference in ONL thinning in the ABCA4 knockout mouse, we went back to measure iron levels in these mice. And we found that in RPE cells in ABCA4 knockout mice, levels of iron were higher. This is here we used on the left a colorimetric assay. Ferrozine is a chelator that acquires a magenta color when it's bound to iron. And so this enabled our detection. And we found, as I said, that the iron levels were higher in the ABCA4 knockout mice RPE. Josh Danaev's lab also immunostained for ferritin. Ferritin is a storage protein in cells, and it's a very good indicator of iron levels. And in the ABCA4 knockout mice, the ferritin immunostaining was increased, again indicating that in the ABCA4 knockout mice, ferritin levels and therefore iron levels are increased. Josh then also had sections from individuals diagnosed with recessive Stargardt's disease and stained with an anti-ferritin antibody. Here you can see on the left the no primary controls, and on the right, anti-ferritin labeling in the healthy eye on the top and in the recessive Stargardt eye on the bottom. So there was an increase in ferritin in human eyes diagnosed with recessive Stargardt's disease. So I certainly don't think that this increase in iron is in any way a primary factor in the disease, but I kind of wonder if during the degenerative process, perhaps when RPE cells are stressed, perhaps the control of iron levels is not sufficient. I'd be happy for anyone's input when we have time for questions. So I'm going to shift gears a little bit now and discuss some of our findings related to studies of fundus autofluorescence. In recent years, working with Francois Delory, we developed a method for that we call quantitative fundus autofluorescence. I don't have time to go into it, the details, but just to say that we adjust gray levels according to this an internal fluorescent reference that is mounted in the Heidelberg Spectralis. And we acquire measurements we call QAF values at a position 7 to 9 degrees outside the fovea. So it's in this ring here on the left that is bolded. And that's where we acquire our values. So first we found that RPE bisretinoid light perfusion increases with age. And this was based on quantitative fundus autofluorescence measurements. Of course, this was not new. This was information that was already available, for instance, from histological studies over many years. What QAF offered us, however, was some spatial information. Also, we noticed that the range of values really increased in healthy subjects after around age 20. And these images on the right here are what we call QAF color-coded images, where pixels are assigned a color based on their gray level intensities. And you can see from individuals age 6 through 42 years how the intensities increase, with blue being lower intensities of fluorescence and yellow and red being higher levels. We also found that QAF was increased in recessive Stargardt's disease. This also was not new. It had been reported from histological studies and also from spectrophotometric studies of recessive Stargardt's disease. On the left here, you can see the red lines are the mean of our group of healthy eyes, with the confidence intervals indicated by the dashed lines. And the values for the recessive Stargardt patients are the blue circles. And you can see that in most cases, QAF is increased in the individuals diagnosed with ABCA4 related disease. Again, though, there's considerable variability. And in these QAF images, again, you can see these are similar aged individuals from age 20, 25, 19, and there's considerable, this is the healthy eye on the left here, sorry. And even amongst the recessive Stargardt patients, there's considerable variability in these images. And of course, we know from the work of many, many groups, we can understand what's going on here with ABCA4 deficiency. As I mentioned earlier, both 11-cis retinal and all-trans retinal can react to with phosphatidylethanolamine in outer segment discs, forming this shift base, NRPE. If NRPE hangs around and reacts with a second molecule of retinaldehyde, it will go on to form bisretinoids. And of course, I should also mention that NRPE it is in the form of NRPE that retinaldehyde is delivered to the cytosol by ABCA4 so that it can be reduced to the less reactive retinol, all-trans retinol. In the ABCA4 knockout mouse, we found that this mouse replicated findings in the human subjects. The blue here are the fundus autofluorescence measurements, QAF measurements, and the red are HPLC measurements. And you can see with both approaches, bisretinoid levels are increased in the ABCA4 knockout mouse. We also find that in the ABCA4 knockout mouse, in the albino, outer nuclear layer thinning occurs and can be observed by about 8 months of age. And you can see in the plots below here, the red is the wild type, the blue, the ABCA4 knockout, and they're thinning at age 8 to 9 months. And on the right, the thinning is progressive with age. In the sections above, you can also see on the right is the knockout mouse. You can readily see thinning as compared to the wild type on the left. So we also have been interested in QAF and its ability to help with some initial phenotyping. It can be difficult to draw relationships between genotypes and phenotypes in recessive Stargardt's disease, as all of you are aware, because there are so many mutations and the frequency of compound heterozygous mutations is great. But in a few instances, we can try to make some comparisons. And one example is with this, comparing this complex allele here with the mutation G1961E. So with this complex allele, you can see that QAF levels are high at a very young age. This was these are from approximately a 10-year-old. And also you'll note from the QAF maps on the right that there's extensive retina-wide disease, even at a young age. Compare that to an individual here, the QAF map is below on the right, of a G1961E individual. Here, the G1961E is well known to confer a bullseye maculopathy. So the disease is confined centrally. You'll recall that I told you we do our measurements at 7 to 9 degrees outside the fovea. And at that position, we don't measure, there is no increase in short wavelength autofluorescence. There's no increase in QAF, and you can see that the values for the G1961E patients are often within the normal range, shown here with the gray lines. I'm sorry, I'm doing that by mistake. And so with the G1961E, the values are very often within confidence intervals of healthy eyes. So it would also seem that the disease correlates spatially with the increase in bisretinoid that is detected by QAF. Here we did a study where we studied, where we looked at a number of patients that presented with bullseye maculopathy, some of which had ABCA4 mutations, and others that did not have ABCA4 mutations, but presented with a bullseye phenotype. And we compared their QAF levels. Here, on the left, you can see the mean and confidence intervals for healthy individuals. The blue squares are the non-ABCA4 individuals, and they mostly fit within the confidence intervals of healthy eyes. While in the presence of the ABCA4 mutation, most of these individuals had relatively very high levels of QAF and at early ages. So QAF kind of helps to segregate groups of patients that may even present with similar phenotypes. We also looked at QAF as relates to ABCA4 carriers. There had been debate for many years that as to whether ABCA4 carriers could be responsible for late onset disease. And thinking, of course, that an important component of the disease is our bisretinoid levels as measured by QAF. We examined carriers and their probands. So the symbols that you see here that are filled are the values for affected ABCA4 positive individuals carrying 2 mutations. So these are the recessive Stargardt patients. The symbols that are only half full are the values we obtained for the carriers. And you can see that in most cases, the carriers, the values for the carriers were within the normal limits. The normal limits determined for healthy-eyed individuals. So on this basis, we concluded that ABCA4 carriers do not express increased levels of short wavelength autofluorescence. I should point out that this had also been reported by a group of individuals that included Peter Issa, Bernard Weber, Frank Holtz, and Robert McLaren. But actually, QAF became very interesting with respect to another set of carriers. These are images we acquired from two siblings, age 15 and 13, that carried these mutations in RLBP1, which encodes for the visual cycle protein CRAL. And the mutations were this missense and a deletion. Now, I'll just remind you for a minute about CRAL. Of course, to regenerate the 11-cis chromophore, RP65, we all know, carries out this isomerization, and CRAL assists RP65 by binding the product 11-cis retinol as it's being formed so as to drive the reaction forward. And also by binding the 11-cis chromophore, it maintains the 11-cis configuration. So it's it's not necessary for the isomerization, but it certainly plays a role. So back to these siblings, we found that they presented with very reduced fundus autofluorescence. The short wavelength images are above and the QAF images below. Very reduced QAF, but with this speckled fluorescence centrally. This group of patients, the CRAWL carrying CRAWL mutations, are part of what has been described as the white dot diseases because of the speckling that is observed in images. You can see that there was also fairly broad hypertransmission of the OCT signal, indicating problems in the RPE monolayer. And we observed hyperreflective foci in outer retina. Looking at a RLBP1 knockout mice that we obtained from Vladimir, we found that these mice replicated what was being observed in that family. 11 cis retinal was decreased. Short wavelength fundus autofluorescence measured in the mice was reduced relative to the wild type. And we observed a thinning of outer nuclear layer. You can see in the sections here, the wild type is on the bottom. You can see the big difference in the rows of outer segment of outer nuclear layer photoreceptor cell nuclei, and the measurements of nuclei per column are above. So there was thinning of the ONL indicating a loss of viability in the photoreceptor cells. But it got really interesting actually when we looked at QAF images of the parents. The carriers of RLBP1 have been considered to be without symptoms. And in fact, the parents reported no visual problems. Their images on short wavelength, near infrared were all normal. Similarly, the OCTs were all healthy. You could see the father carried the missense mutation and the mother the deletion. But when we looked at the QAF images, it was clear that there was a phenotype. On the far right is a QAF image from an age-similar individual. And looking at the QAF images of the father and the mother, there's clearly reduced signal in both of these individuals. And the values are plotted on the right-hand side. In both cases, the values were below the confidence intervals for healthy individuals. To us, this indicated probably a kind of chronic long-term reduction in the visual cycle. There's also a case of RPE65 carriers that I'm going to show you. In this family, the proband short wavelength autofluorescence in the program was not recordable. I have QAF images here of the father and of the mother. And in most, it's generally considered that RPE65 carriers are symptomless, except in one case of a dominant variant. But we found that, and you can see the mutations that that the mother and the father carried, and we found that in both cases, well, I should say, in the case of the mother, it was clear that QF was reduced. And the plots of their values are over on the right-hand side. You see for the mother, the value was below the confidence intervals for healthy eyes. In the case of the father, he was within the normal range, but in the lower limits of normal. So there can be, depending on the variant, there could be for RPE65 also sort of chronic, maybe long-term, a slowing of the visual cycle that is not necessarily detected by in terms of other symptoms. QAF is also reflects vitamin A deficiency. This was an interesting case of a male aged 61 years that had bariatric surgery 13 years earlier. The serum vitamin A levels were low. We also recorded low QAF. You can see the image here. The second from the left is the QAF image we acquired at his first visit. On the far right is a QAF image from a similar aged healthy eye. And so you can see that his QF was quite reduced relative to what you would expect. He was given intramuscular vitamin A injections weekly over 3 months. And his QAF image after vitamin A treatment is below here. When I saw this image, I said, wow. With this high dose vitamin A, he had acquired fundus autofluorescence that was very, the levels of which were very similar to what you might see in a healthy eye of a similar age. This is what was different, however, and I hope you'll notice, is that the topography was different. In the healthy eye on the top right, you can see that the QF intensities are greatest superiorly and temporally. In this individual that had received the high vitamin A dose over 3 months, actually the highest levels were more inferiorly. This suggested to me that after the formation and deposition of bisretinoid in RPE cells, there must be some refinement of the topography. And one way in which this refinement could occur would be by light-mediated photooxidation and degradation. I'll point out another interesting feature of his images. He had very low levels of macular pigment. This is not surprising if because of bariatric surgery, he had very low levels of serum vitamin A. It wouldn't be unexpected that his carotenoid levels would also be very low. And in fact, that's what was observed. He had very low levels of macular pigment. And in his post-treatment image, you can see he gained a lot of fundus autofluorescence, but he still had little or no macular pigment. In the next few minutes, I'm going to talk about some cases in which we don't really understand why there should be increased fundus autofluorescence. So in the case of ABCA4, of course, we all know the pathway. It's been well worked by many, many investigators. The same would be true for deficiency in retinal dehydrogenase. We can appreciate why increases in bisretinoid might be observed. However, there are some cases where we think that maybe increased bisretinoid could be a secondary feature of photoreceptor cell degeneration. One example here is in peripherin 2 RDS. So perforin 2, known to everyone, is a structural protein of outer segments. Perforin 2 associated disease is also autosomal dominant, manifested midlife, and it has a range of phenotypes. And so these RDS patients are considered to be grouped within the pattern dystrophies. So we acquired QF images and quantitation in some of these individuals. The perforin RDS positive patients are the blue circles. And you can see that in some cases, the perforin RDS, some cases of the perforin RDS positive patients, the QF levels were high. Other cases, of course, they were within the normal limits. But we were interested in the fact that in some cases, these could actually be as high as in ABCA4 positive individuals. And perforin RDS, of course, presents with disease that is readily observed in fundus autofluorescence images, atrophy, modeling, flex. And in the OCTs, again, you can see hypertransmission of the OCT signal into the choroid. You can see flex, a lot of outer retinal degeneration. So we don't readily have an answer as to whether or as to why in these RDS patients, one should see an increase in fundus autofluorescence. There are other examples also of our observation of increased QAF in retinal disorders. For instance, I have some images here of acute zonal occult outer retinopathy, or AZUR as it's called. The line between the lesion and non-lesion retina has been called the AZUR line by Larry Iannuzzi. And we can sometimes see, as is visible in the image second from the left, that there is high fundus autofluorescence along that AZUR line. These images of retinitis pigmentosa will be familiar to many of you. It's fairly common to see these autofluorescent rings in RPE. These rings are not specific to any genotype. They can be found in both dominant and recessive disease, and they've been well studied and are considered to reflect a transition between healthy and unhealthy retina, with function being absent outside the ring, being reduced over or within the ring, and being normal inside the ring. We found that in about 30% of cases, this autofluorescent ring, hyperautofluorescent ring, was an actual increase in autofluorescence as compared to a healthy eye of similar age. So this was an increase in short wavelength autofluorescence at the position of the ring when we compared it to the fundus of similar aged individuals. So I don't have time to just present other cases, but suffice it to say that we've observed in these cases and in others cases that there can be an actual increase in short wavelength autofluorescence and therefore in bisretinoid production. And this increase is probably stage specific. My interest is in understanding what's going on with photoreceptor cells under these conditions. Why is this increase in fundus autofluorescence? And my interest is in also trying to understand what's common to all of these disorders. And so I'm going to end with a question. Is it possible that when photoreceptor cells are stressed, when photoreceptor cells are impaired due to a variety of conditions, that the photoreceptor cell is no longer able to handle retinaldehyde efficiently. And therefore, one finds that there's an increase in this bisretinoid formation because of unchecked retinaldehyde. Okay, just quickly some points that I've made, that bisretinoids form the lipofuscin of retina. They're also the source of fundus autofluorescence. They play a role in recessive Stargardt's disease. They can undergo oxidative processes, both due to light exposure and iron catalyzed production of hydroxyl radical. And I suggest that the amount of accumulated bisretinoid at any given time may not be as significant as the total amount formed and the amount lost by oxidation. In Merticate deficiency, bisretinoids account for the autofluorescent debris that accumulates. And QAF may be able to inform us about carrier status in some cases, for instance, ABCA4, RLBP1, and RPE 65. Thank you very, very much for your attention.
We have already a few questions ready to be asked, and let me start, Janet, and the good news is we have Gabe who is going to make a final comments at the end. So my question to you is about the MRTK. As you know, we've done earlier study on MRTK, and unequivocally, the A to E is present in those lines and quite abundant. And we can do this by 2 photons. So there's really a lot of evidence, chemical evidence that is present. So it must be a different mechanism that also RPE is getting this retinoids. And so good news is also we have Roxana here. So there's a lot of also ideas of making from 11 cis retinal, these retinoids in RPE. Can you comment on that?
Yeah, so I think that I agree with you that there could be other mechanisms, other routes, or not routes necessarily, but other reasons why bisretinoids could form more than just more than those mechanisms that we now can now appreciate or understand.
Vladimir.
Hi, Janet. Thank you so much for a thought-provoking presentation. I have many questions, but I'll ask a couple. First, I should comment that the CRLBP mice that you kindly acknowledged us, they were actually Jacques Saris mice, so we just passing them along, but those are Jacques Saris mice. Okay. So the QAF measurements that you have done, first, do you see any evidence for, or do you know if they are dependent on the state of light adaptation of the patient or of the animal? And more broadly, when you have done all of those measurements in patients or in mice, do you see any evidence? Is the fluorescence limited only to the RPE, or do you also see evidence for maybe change in fluorescence in the neural retina?
Yeah. So your first, So your first question was, yeah. So when we do the fundus autofluorescence measurements, we typically use or always use light adapted mice. And I don't think that measuring fundus autofluorescence is going to be affected by a short-term dark or light adaptation. So we don't necessarily worry about that. But in practice, actually, the mice are always light adapted. So measuring fundus autofluorescence is not the same as, for instance, measuring recovery of 11 cis retinal after bleach or measuring dark adaptation. And then your second question, I'm really, really sorry. What was?
Whether the fluorescence is limited to the RPE or you see
yeah.
changes.
Yeah, this is a very, very interesting question to me. I can't really answer the question with any certainty. I certainly think we have actually observed in some cases, even in a non-human primate, we observed autofluorescence that was located in outer segments. And that was in a healthy eye, or supposed to be a healthy eye. So I don't know. I think that although we haven't had a lot of occasion to examine this question, I think there should be examples whereby you can see the autofluorescence in photoreceptor outer segments and even inner segments. In a diseased eye, even in inner segments.
Okay, thank you.
Arthur.
Hi, Janet. A wonderful presentation. Thank you. I have a comment and then a question. The comment was you mentioned RP65 carriers and possibly having a chronic slowing of their visual cycle. That reminds me back 22 years ago in a collaboration with Michael Danziger and Deborah Farber, we had published careful dark adaptation kinetics in two carriers, carrier parents of an RP65 patient. And just for your information, they were perfectly normal, both after small as well as large bleaches. So at least psychophysically, looking at the kinetics, there was no evidence that those two mutations were affecting the recovery in the short term. Of course, chronically, what happens is a different question. But the part that I wanted you to comment on is you have a wonderful literature on near-infrared autofluorescence. You didn't touch too much on it here, and you did mention the photoreactions in vivo in patients that the bisretinoids may be having. Would you comment on the possible interaction of changes, specifically increases of melanin-related signals, and how that could potentially interact with any of the photoreactions in a real patient with real light?
Mm-hmm. Yeah, I initially had included near-infrared autofluorescence, but it was just going to be too much, so I actually cut out images, except in one case. Yeah, so with respect to near-infrared autofluorescence, we've done a lot of imaging, and we find in both ABCA4 positive individuals and in ABCA4 knockout mice that the near-infrared autofluorescence images intensity is increased relative to the amount of bisretinoid. So what I mean by that is that in a Stargardt patient, near-infrared autofluorescence is going to be increased relative to a healthy eye. And that's true. That's true also in the mouse. That's also true in the mouse. And we don't fully understand it, but we think, for instance, if perhaps in the case of near-infrared autofluorescence, if there's a lot of lipofuscin granules that are separating the melanosomes, the melanin granules, maybe more of the emission can leave the cell rather than being absorbed locally. And that's about the only explanation that I can give. I know people have talked about lipofuscin increasing melanin synthesis, but I personally, I don't think that's what we're looking at.
Hey, John.
Hello, thank you so much for the talk. I was just wondering if, looking at these QAF images, have you ever done any studies of like a longitudinal study where fundus autofluorescence was observed over time in an individual with a certain mutation or carrier allele to see if the autofluorescence changes kind of align or correspond to the disease progression. And also, it seems bisretinoids have, my second question is, it seems bisretinoids have either a marker, can serve as either a marker of visual cycle status and health or metabolic impairments. How is it best to discriminate the role of bisretinoids in or deconvolute that dual role that they kind of can serve as a marker of either?
So in terms of metabolism or a metabolic marker, I know this is often stated in the literature that they're a marker of metabolic impairment in RPE cells, but I don't agree with that for a couple of reasons. One, because they form in photoreceptor cells, and two, as I tried to explain in some of my early slides, that there's really no evidence to conclude that there's a problem with lysosomes in RPE cells that are accumulating bisretinoid. Part of the reason I say that is that, you know, we all, even healthy-eyed individuals, we all accumulate bisretinoids. And I don't think the problem is with the lysosome. We're not, you know, we're not turning into lysosomal storage disease or something like that. I think that these molecules, they are forming randomly. There's probably no design for them, and therefore that the structure just may not cannot be attacked by the normal complement of enzymes in lysosomes. That's my opinion.
All right, Patricia.
Hi, Janet. Great talk. Thank you so much. My question has to do with your first part of your presentation with regard to your bisretinoid phospholipids. I'm curious about PLA2 and PLD. Do you know the type of PLA2 that would act on your bisretinoids A2Es? And also if you've used any inhibitors or anybody has used inhibitors to PLA2 to identify this reaction. If we could prevent that reaction, you will have less A2E, correct? So anyways, the question is about more information about the PLA2.
Yeah, the PLA2, actually at this moment, I can't give you any more information. We tested it in vitro with PLA2. We have not used an inhibitor. With regard to PLD, we have used inhibitors of PLD. We've used the commercially available enzyme to show that it does act on that phosphodiesterase bond. We've also subjected A2PE to extract of liver lysosomal enzymes and observed that cleavage. And we've used inhibitors, as I said, we've used inhibitors of PLD. I don't know if that's helpful.
Yeah, well, we're working on a PLA2. Perhaps you and I can have a conversation on the side about it.
Sure.
Yeah, thank you.
So we have last two questions before we're going to Gabe for final comments. Dominic and Zach.
Hello, Dr. Sparrow. Great talk. Thank you very much. I have a question regarding this accumulation of ferritin in RP in the recessive Stargardt patient. Did you consider maybe that this could be some kind of a protective mechanism and not the detrimental one? Potentially, since macrophages are by producing pro-inflammatory cytokines like interleukin-1 beta, IL-6, and TNF-alpha, stimulating ferritin expression. So did you observe also any inflammatory events in this patient? Like, yeah.
Yeah, that's a very good question. We haven't done those studies. And certainly the amount of L-ferritin is sort of reflects the iron levels. I agree with you that if there's iron around, the cell will want to synthesize more ferritin in order to store it. But I'm not really sure what else I can offer because we haven't done any of those studies. And you know, this could be the fact that there's increased iron could be like way downstream of some of the disease features.
All right, Zach.
Thank you, Dr. Sparrow, for that really amazing talk. That was kind of a good preface to my question, which also involves increases in iron and ferritin levels in the ABCA4 knockout mice. I was wondering if you see any like concurrent changes in antioxidant levels or oxidative stress markers that might imply some sort of change in metabolism.
Yeah, again, we haven't done any of those experiments to know. Certainly there's a lot of evidence that antioxidants protect or attenuate photooxidation and degradation of bisretinoids. But in terms of the specific experiments you're asking about, we haven't done them.
Thank you.
All right, so next week we have Rendo Alikmets. Please join us for that presentation. Invite your friends, enemies, neighbors, and local politicians. And now I'm giving you Gabe.
Okay, can people hear me? Yeah. So I wanted to just add kind of a couple of thoughts. Kind of concerning the function of ABCA4 or I should say maybe functions of ABCA4. And this is actually prompted Dr. Radu, who's here and probably will comment, and myself earlier, but now she's taken over this project. There's an observation that Janet mentioned, which is that actually you don't see increase, you might actually see reduced bisretinoids in light exposed animals. And according to the model that we saw in terms of the ABCA4 function in the photoreceptor outer segments, this doesn't really make sense. Because the source of the A2E, as explained, would be, or let's just say bisretinoids, should be come from photobleaching of rhodopsin and release of all-trans retinaldehyde. However, that doesn't seem to be the whole story. So in a paper of few years ago now, I don't remember exactly when, Roxana Radu showed that ABCA4 is also expressed in retinal pigment epithelial cells, and this has now been repeated over and over by multiple people. I don't think anybody who's looked for it has not found it, certainly at the RNA level. So the question is, why do we need ABCA4 in the RPE? And I think the idea that was put forward is that there are really two processes here. The one process is photobleaching of rhodopsin in the outer segments due to light, which releases toxic all-trans retinaldehyde and has to be dealt with. And one way to deal with it is to get it out of the outer segment and into the, get it out of the bilayer and into the cytoplasm of the outer segment where it can be reduced to vitamin A. And that is something that, that ABCA4 obviously is participating in. But the other thing which we don't always think about is that there's sort of another visual cycle, and that's through phagocytosis. So we have, as we know, 10% of distal outer segments in rods and cones are phagocytosed every day. And that means that approximately 10% of the total retinoid content of the eye is passing through the RPE through phagocytosis of the rhodopsin in the outer segments. So what's the fate of that? Well, we know that the outer segments are degraded, proteolyzed and everything. And so every rhodopsin molecule releases a retinaldehyde. And another possibility is that this retinaldehyde has to be dealt with for the same reason it has to be dealt with in photoreceptor outer segments, and that ABCA4 may be doing exactly the same thing in the photoreceptor outer segments and in RPE cells. And this would at least explain why we wouldn't see a difference with dark or light-reared animals, since phagocytosis continues regardless of light exposure history. And I think I'll just leave it at that for now.
Right. So both. Both the group in South, I may have misunderstood you, Gabe, but the group in South Carolina and our group has shown that even in the dark, bisretinoids form, and so that it seems that the source would be 11-cis retinal when the mice are raised in the dark, correct?
Well, the fact is, as soon as the rhodopsin is degraded, undergoing proteolysis, I don't think you could say it's going to remain as 11-cis retinaldehyde. I mean, it's going to be, I mean, those are interchangeable. But yeah, okay.
Yeah, I agree with you. A retinaldehyde that started out as 11-cis could subsequently contribute, right? And the amount of ABCA4 in RPE expressed by RPE cells was at about 5% of the total.
Yeah, it's quite low, but at the same time, if you think about it, there is need for much more ABCA4 in the outer segments, where you have ongoing, where the visual pigments are being bleached and the retinaldehyde is being released all the time, and that has to be continuously processed. The amount probably required and the amount of membrane surface area is vastly smaller in the RPE, whatever structure that is in, some kind of internal membranes probably derived from lysosomes. Yeah. So I don't think we're bothered by the fact that it's lower levels in the RPE. Are you?
Oh no, I'm not. No, I just thought I would clarify.
Okay.
Do we know in a phagosome of outer segment, do we know what the relative amounts of bisretinoid versus retinaldehyde in those phagosomes?
Roxana might be better at answering that question. I don't know the answer.
So, hi, Jana. I really enjoyed
Hi,
your
Roxana.
talk. Very nice data. The relative amount of retinoids obviously depends on the status of light when they get internalized. So that's kind of difficult to say. But I just want to clarify this idea that through those phagosomes, you have this pool of free retinaldehyde that are released in the RPE. And as Gabe mentioned, those aldehydes have to be dealt with right there. So what we put forward is just, sort of initiated a dimerization process that takes place in the RPE. And we clearly addressed this experimentally with different models. So one was in vivo, where we express ABCA4 in the RPE only because that transgenic mouse was done on a null background. And there, clearly, the presence of the ABCA4 in the RPE only could significantly reduce the level of the autofluorescence, and obviously that also correlated with the levels of bisretinoids that were quantified by HPLC. So without being safe, so that's a living animal. But then we follow up with a different model that obviously that took a long time to build this iPS-derived RPE cells from a normal individual, and from Stargardt individuals. And they are in culture, so those cells has never seen outer segment till we provide those outer segment and monitor, the recycling of those retinaldehydes. And clearly the cells that have a mutated ABCA4 are unable to clear those retinaldehydes. And also those cells shows autofluorescence increase over time. So, I mean, that it's like recapitulated key phenotypic features of Stargardt disease in an RP only model. So that kind of goes with that. So there is an endogenous bisretinoid formation, or at least not just bisretinoids, a process of creating those in the RP only. And that clearly is dependent on ABCA4 in the RP. So those are models, in vivo and in vitro that speaks to this, endogenous new dimerization formation. And the source must be aldehydes from the outer segment internalized, or any kind of free retinaldehydes that are in the RP. That's kind of the simple modality of putting this process. So it's new. And I think the aspect of ABCA4 being in RPE and modeling two different modalities, I think that speaks volume. And I would like to make a comment regarding this secondary effect that you describe in other disease that are the pure photoreceptor disease, because that's where the genetic mutation is. That may also reflect that internalization of those outer segment following phagocytosis. It's possible that it's a stressor for the RPE, and the way that RPE deals with those free aldehydes released following the proteolysis of that rhodopsin may indicate the existence of this pathway of dimerization formation in the RPE. And actually, furthermore, like Chris started with MERTK, I think that's what happened, and also along with Gabe's comment, it's internalization of the phagosomes or outer, it can be done independent of MERTK protein, but the resulting is you have a material containing free retinaldehyde, and that itself leads to increased autofluorescence.
Yeah. Yes. And that's
I mean, there are many arguments to support this, bisretinoids formation in the RPE with free aldehydes, aside of the pathway that you described and many others describe. And I definitely support that. So dimerization formation in photoreceptor membrane for sure take place without doubt.
All right, we had a wonderful conversation. This will absolutely continue unless Vladimir or Janet wants to make a final.
Can I just make a quick comment to the issue of phagocytosis through RP as another visual recycle mechanism? So my former colleague at WashU, Tom Ferguson, showed a few years ago that autophagy actually in RP specifically plays a role in this process, and mice where autophagy was blocked specifically in the RP had chromophore deficient rods, and their function could be restored with exogenous chromophore. So it seems to me that that might be one model that might be useful to get to the bottom of this question, because that will allow you to specifically block the recycling of chromophore through the phagocytosis pathway, and then you can look at changes in bisretinoids and overall visual cycle. So that's my comment.
Thank you, Vladimir.
Janet, thank you so much for joining us. Unless you have any final thoughts, we will continue. It looks like a very important area of research.
Thank you very much. Thank you, everyone.